Great. Thanks everyone for coming, and thanks to Professor Scher for inviting me. I'm really excited to be here today to talk to you, and I'd love for you to interrupt me with questions anytime and make it interactive, just make sure that everybody's following along and good with what we're chatting about. So here's a brief overview of what I'd like to go through with you. We're going to have 3 different parts that we'll go through. The first is about how genomics is currently used in healthcare. So this is kind of a landscape of the current state of affairs and gives you a sense of where things are, and then we'll talk just a little bit at the end of that section about where we're trying to bring things now with our current work. Next, I'd like to go into what is DNA, how do we study it. This is the basics of the current technology, and we'll talk a little bit about the ways that we're working to improve the technology. And then the last part is about what can we do to expand the use of genomics in healthcare. So this is really the work that I've been part of at the Genome Center and now at Rockefeller University. All right, so the first section is going to be about how we use genomics in healthcare today. So the first, basically I have several different examples of ways that we can use it now to give you a flavor for how your DNA can be interpreted in terms of your healthcare. So the first one is sickle cell anemia. So this is a classic textbook example where there's just an extremely minor change in an individual's DNA. And you can see in this, hopefully you can see in the photo here that what happens is the shapes of your red blood cells, instead of being sort of like a donut shape, they end up being a sickle shape. And they end up piling up in blood vessels. Okay, and so this is a very minor change in the molecule that carries oxygen in your blood. And what happens is the cells get stuck together, and it basically can be like a blood clot where the blood can't flow through anymore and you can't get oxygen to your tissues. So what you have to do if this happens is go to the hospital and get treated right away. If you don't, then you can end up with organ damage, and it's very painful whenever it happens. So this is a really severe disease from just a tiny change in the DNA. And this is primarily in African American population. So 1 in 13 African Americans are carriers of this disease. So they don't manifest it if they're carriers, but 100,000 Americans have the disease. And so this is just, again, a tiny, tiny change that can lead to something that causes extremely dramatic effects that can really affect your lifestyle. Yes?
I have a question. Is this genetic?
Yes, it's genetic. Yes. So in this case, understanding the change in the DNA that leads to this disease helps us understand the pathogenesis of the disease. Basically, if someone just came to the hospital saying, I have this pain, you wouldn't know how to treat it if you didn't know what was behind, what the cause was, basically. And it also helps us diagnose the disease. So now if someone comes to the hospital with that sort of pain, you can test their DNA and see if they have this defect, and then understand where it's coming from. All right, another example that you may have heard of is BRCA mutations, sometimes pronounced BRCA, and cancer risk. So this is a figure from the National Cancer Institute that shows a woman's risk of getting breast cancer before the age of 75. So on average, it's about 12% of women will get breast cancer before 75. And if you have certain mutations in these BRCA 1 and 2 genes, it can increase your risk to as much as 65%. Okay, so this is just a major risk that people would want to know about. So there are thousands of different mutations that can occur in these 2 genes, and we're really still learning how to interpret them. There are some that we know well from studying different families, seeing how frequent the breast cancer development rate is, and there are others that we're still learning about, and this is really an active area of development. So another thing to think about is, we know that these mutations can increase your risk of getting breast cancer, but actually only 5 to 7% of women that have breast cancer have one of these mutations. So there are many other ways that cancer can arise, and it is a primarily genetic disease, and I have a little bit more about cancer and genetics later in the presentation. And even in the hereditary breast cancer cases, only 20 to 25% of the women have a BRCA mutation. So another thing that you may have heard about related to BRCA is that Angelina Jolie found out that she had one of these mutations, and she decided to have a mastectomy, basically removing the breast tissue before she developed cancer. So knowing about this mutation allowed her to make a proactive choice to avoid the disease. So that's kind of the most dramatic way to be preventative about the disease, but you can also, if you know that you have these mutations, you'll have more frequent screenings, like every year or so. And your insurance company will know that that's what you need, and it just allows you to be more proactive about your health than the average person would need to be. All right, so knowing about this change in the DNA helps us anticipate the disease with the Angelina Jolie type example, and it also helps you treat the disease. So there are certain types of drugs called PARP inhibitors that are more effective in women where we know that this BRCA gene is mutated and that we think that's the cause of the disease. So it helps us, again, understand and treat the diseases in addition to being more proactive about trying to prevent it. Another interesting area where genomics can affect you is drug metabolism and different side effects. So basically, everyone can respond to drugs differently. You've probably had this experience where you know that a certain drug might work for your friend, but it doesn't work for you. So I have a few different examples about ways that the drugs can affect you differently. So this is a list of different drugs, the conditions that they treat, and I'll talk a little bit about the effects that they can have. So simvastatin is a very common drug for treating high blood pressure. And what happens in some individuals, a very small subset, they can develop an irreversible muscle weakness from taking this drug. Okay, and this is something that, when it arises, it can happen, years after you start taking the drug. And it might be really hard for you to correlate it in your mind and say, oh, this is a result of this drug. Your physician, it might be difficult for them to figure it out. And so if you don't stop taking the drug soon enough, you can end up with irreversible muscle weakness. However, if we tested people's DNA ahead of time before they got this prescription, we could prevent these side effects in the small number of individuals that would get them. Another drug is codeine, which is used to treat pain and cough. And so what happens in different individuals, either they metabolize it too slowly, and so basically it lessens the dose that their body is able to process over time, and then the drug is not effective for them. Or they metabolize it too quickly, and then they have more of the drug in their system at a time, and it can lead to toxicity. So knowing whether you're on one of those extreme ends of the curve would make your doctor make a different recommendation for you related to this drug. Allopurinol is used to treat gout, and this one can lead to adverse skin reactions that actually can be potentially fatal if you don't catch it at the right time. So this is another one where your doctor would just prescribe you a different drug. And the last one is a class of drug called thiopurines. They're used to treat arthritis and cancer. And this one is a really extreme side effect. The normal dose is fatal on people that metabolize it in this extreme way. And so actually, it's mandated by the FDA that you test for this mutation before you prescribe the drug, just because it's so dramatic. So actually, the individual can still take the drug, they just take it at a 10-fold lower dose than a normal person. Okay, so it's just, can be so dramatic how differently you metabolize the drugs. So knowing about these changes in DNA helps you manage the disease more effectively by making sure you're prescribing a drug that will work for this individual, and it also helps you avoid negative side effects. All right, and then my final example is about familial hypercholesterolemia. It's kind of a mouthful, so we call it FH for short. And individuals with this disease have naturally high cholesterol levels. So this is something that, again, can be kind of hard to diagnose because we know that some individuals have high cholesterol levels because of lifestyle choices. But for these individuals, even if they have just a normal lifestyle, their levels are extremely high. So actually, it ends up with causing a lot of early heart attacks in families where individuals have this disorder. And so as soon as the doctor figures out that they have it, they put them on a low-fat diet. They cut out things like egg yolks to keep their cholesterol down. And they see if that's enough to lower their levels of LDL, which is the bad cholesterol that you're not able to eliminate from your body if you have this disorder. So if that doesn't work, then they go on medication. One of the medications that they use is simvastatin, so you want to make sure they don't have the simvastatin issue and get into muscle weakness problems. And this trait is actually dominant. So what it means is you only, we'll get into the chromosomes a little bit more later, but basically it's easier for you to get this disease. And what happens is if they find one person in the family that has this trait, because it's so underdiagnosed, what they do is they do what they call cascade screening, where they do, everyone's looking down, great. So they do, they test everyone that is in the immediate family of that individual, their second degree relatives and their third degree relatives, because it's so likely that all these individuals are also undiagnosed and have this disease, and they can be proactive about managing it. Yes, question?
Is the condition primarily found in females or males, or it has no correlation?
No correlation. For this one particularly, there are some that are linked, like things that are on the X chromosome are found more commonly in males because since females have two copies, if they have one functional copy of the gene on one of their X chromosomes, then they could often compensate for the allele that, or the mutation on the other copy of the X. But since males have one X and one Y, if their X copy has a mutation in it, then that's the only one they have to deal with. Colorblindness is one of the traits that's linked to it. Good question. Right, so in this case, knowing about the disease, or knowing about the change in DNA helps us, one, diagnose the disease in the individual that's at the doctor's office at that moment and in their family members. And this is actually something, since it can cause heart attacks so early, they really are trying to push to have it diagnosed in children, because once you get so far in having your LDL levels build up, it's very hard to go back. But if you can be proactive about the diet when the person's a child, then you can fix it before it gets too bad, if that makes sense. Okay, so currently the state of affairs of all these examples that I mentioned, we use genomics when the person's healthcare at that specific time requires it. And what the group that I'm working in is trying to do is push toward looking at all of every individual's DNA, right? If we had enough discoveries and enough times that genomics was incorporated into your clinical care, it would make cost sense and just healthcare sense to be able to look at everyone at the beginning of their life. We could decide at the exact age, maybe you wait till people are 18, but essentially to just have that data there for everyone is something that we're striving for. So some advantages of that would be we could reanalyze the data whenever we need it. Before your physician is going to write a prescription for you, they could interrogate the literature and see if there's any mutations that would counterindicate this medication and say maybe they should make a different choice. It could lower the lifetime costs for one individual, because one of these genetic tests where they look for one specific mutation can run thousands of dollars. And the cost of whole genome sequencing, where you look at all of the mutations possible in your body, is coming down over time. It's about $1,500 right now. So if it comes down and down and down every year. In a couple years, that cost curve will probably cross where it would make more sense to just sequence everyone up front. It allows preventative action, like some of the examples that we discussed, like writing prescriptions, or in the BRCA mutation cases where you might want to have more frequent screening. And finally, one of the things that we focus on is using large data sets from diverse populations to be able to move research forward. So, all of these discoveries had to be made in someone's DNA where someone had both this genetic defect and we had the clinical information to be able to put the pieces together to make that discovery. And the more data we have, the more discoveries we can make, and then the more value everyone gets out of having their DNA sequenced. That's all for the first section. Does anybody have any other questions on that part?
How much do you think pharmaceutical companies are pushing for this not to happen? Or are they pushing
Not to happen.
for it to
So pharmaceutical companies like it for drug target discovery. So, one, drug target discovery. So things like I mentioned in the BRCA case, they have these PARP inhibitors that they know are used for, or more effective in women that have these specific mutations. Another reason that pharma companies like it is when, if they have a drug that they know, they're going through a clinical trial basically to see if the drug is safe to be used in individuals, and if they have two populations where for one population it's really effective, for another population, let's say they have an extreme side effect like the drug side effects that we talked about, if they can differentiate between those two populations and only prescribe it to the population that it's safe for, that allows them to market the drug, where otherwise they might have to give up on it because it's causing these adverse side effects. Yes?
With the BRCA gene specifically, I've read a lot of stuff about, especially with breast cancer, with the screening that happened way too early and interventions that were not necessary. So do you think
Right.
that there's still a lot that we don't know, and if we
Yeah.
get into the whole public domain can lead to more harm, or
it's definitely a balance that we have to play delicately. I think there are separate parts of the healthcare system kind of figuring out which mutations people have and then making sure that the screening is good. I know there's a lot of research into, is something that you're seeing growing fast enough that, it's going to cause a problem, and they can do sequential screens to see things like that. So it's definitely something to take into consideration, that with more screening, you might have more false positives, and you just have to be more careful about it. Right? Yeah, it's definitely a problem. Other questions? Yes?
When the cost comes out, did you say now it's $1,500 to get a full suite of your DNA analysis? And 3 years ago, was that twice that amount, or how fast does it come down?
So it's basically the company that sells the sequencing equipment that's the current state of affairs is called Illumina, and they essentially have a monopoly on the market. So whenever they come out with a new technology, they essentially make the cost drop by 50%. So about 2, maybe 3 years ago, they came out with a new technology that made everything else obsolete, and the price came from $3,600 down to $1,500 just one day to the next. So they usually make their announcements in January, so we're hoping next January they have something really revolutionary that can bring it down by another third at least. Yeah.
Do you think that people have a right to how information about their personal genome is used, or is that ultimately harmful?
So I'll talk a little bit about that toward the end, but I think it's critical personally for the participants who are donating their samples for research to be involved with how it's used, right? It's really an open area of discussion and research at this point, because a lot of people feel that they just want all their information out there and they really don't care who sees it. There's a project run in Boston by a man named George Church, where he says there's privacy is an illusion. Everyone should just put all their information out there. He puts his social security number out there and his signature and his full genome sequence, and he thinks that's what's going to further research and that's the best thing. Others are more conservative. Maybe they're worried about potential discrimination against them for a certain kind of mutation. Maybe for familial hypercholesterolemia, for some reason people would discriminate against you, and they feel less comfortable with that. And so it's really an important part of the process, I think, and certainly in the realm that I work in of clinical research, we're very careful about that, and walking through with each individual exactly what is going to happen to their sample as part of this study, and they can choose not to participate. It's a little bit more difficult when you get into when it's a company's offering where you're not necessarily walked through exactly what's happening. You have a big sheet of terms and conditions that you want to click through because you know what the product offering is and you're really interested in it. So it's definitely an area that it's up for debate and legislation, I think, to some degree. It could be regulated more, or it could be part of the process where maybe making sure people are more informed about what's happening to their data it could be made part of the process. It's another thing on the internet. All of your data on the internet goes who knows where. So I think it's an issue for society at large in genomics and other areas. The next section is about what is DNA and how do we study it. So each person has 23 pairs of chromosomes. This is a little staining of them. And you get one of each chromosome from each parent. And there's a copy of your DNA in every single cell in your body, and I just think it's amazing. Everyone has 100 trillion cells. You start out as one cell, and it just divides and divides over and over again, and you end up with 100 trillion. So I also think it's interesting that each time the DNA is copied, many, many mistakes are made, 32,000. And you have machinery that fixes the mistakes so that on average only 3 remain. But still, over that many cells, there are many mistakes within your own DNA. This can lead to some defects that we can talk about if you want, but it's just interesting that we think of all the cells as being identical, but they're really not. And then this is a schematic of how we get from the chromosome scale that we just saw down to the single base pair level, which is up on the upper right. So each human genome contains 32 billion base pairs. Okay, and when we talk about genomics, we mean all of your DNA, all of your base pairs. Each chromosome has between 50,000 and 250,000, and it has that double helix shape. And the 2 strands in the double helix are, the bases are paired. So A and T are always across from each other on the strands, and G and C are always across from each other. And so when we study the DNA, we sequence it, and what we mean is we're trying to figure out the identity of the bases at each location. And really, we only have to sequence one strand because we can infer the second strand from that. Okay, and then this is a little bit about how DNA is actually used in your body. So about 1% of the genome consists of genes. Okay, and these genes get copied into RNA, and the copying we call transcription. Okay, and then the RNA gets sort of copied, we'll call it copied, into proteins, and we call this translation. Okay, and the proteins are really what do everything in your body. They digest your food, allow your muscles to move, protect you from diseases, and the shape of the protein really determines their function. And any change in the DNA sequence gets propagated to the RNA, gets propagated to the protein, and can cause a change in the shape of the protein that can disrupt their function. Okay, so that's really how changes in your DNA end up affecting what happens in your body. And I want to talk a little bit about sequencing the DNA, how exactly we study it. And this chart that I'll show, or figure I'll show, is from Illumina, the company that I mentioned that sells all the sequencing equipment. So you start with your piece of DNA, your double helix, and you break it into very small pieces, 150 base pairs at a time. And so you can think of this as taking the whole puzzle of your entire genome and then breaking it into tiny puzzle pieces. And that's the chunk that the technology allows us to be able to sequence right now. Okay, and then what we try to do is put the puzzle back together. So we have these little pieces and we want to figure out where they came from. And you'll see this gray sequence on the bottom is what we call the reference genome. And I'll talk a little bit more about that on the next slide, but basically think of it as a key to the code, kind of the average genome. And because the technology is very good, it has about a 99.9% accuracy rate, which is excellent compared to what it was, say, 10 years ago. But over 3.2 billion base pairs, you're still going to have a lot of mistakes. So we make sure that we sequence each base on average 30 times so that we don't have a mistake and we make the right determination in every place, even with that low error rate. And so we kind of collapse all of those sequences into a consensus sequence that we call as this individual's genomic sequence. And just to give you a sense of the probabilities behind mapping those puzzle pieces to the reference genome, if you just had a random sequence of any 4 bases, if you were to map 40 base pairs to randomly match to the wrong place, the chance of that is 1 in 10 to the 24th. So it's a tiny, tiny number of the chance of mismapping. So even if you allow some errors within your 150 base pairs, you can still be very confident about where you're mapping it to in the genome. And then this is a little bit about that reference genome that you use as the key to your code of where to put the puzzle pieces. So it's a single consensus interpretation of the genome right now in its current incarnation. And the current version is based on 13 different individuals that they kind of piece together into one consensus. And right now, about 99% of the genome is included in the reference genome. So there are some areas that are still not accessible. So ways that they're trying to improve it now is they're actually working to make different versions for different ethnicities, because we know that different populations were, isolated from each other, and so different mutations that just arose randomly are now spread throughout that population. And what is, the average appears in the average person's genome in a person of one ethnicity is different from someone in another ethnicity, and that's important when you're going to interpret the variants and deciding, is this something abnormal or not. So that's a current ongoing project, and they're also working on new sequencing technologies that resolve hard-to-read areas. So there are some really important sequences that are in this 1% that are clinically relevant, and so they're working on different technologies that we can talk about if anyone's interested. So next I want to talk about, okay, you have your sequence that you decided is what this person's DNA is, and now you want to compare it to the reference and decide, okay, what's abnormal in this person? So we call these variants, anytime that you have something that's different in your genome versus the average. And there are many different kinds, so I just wanted to show you a few of them. The first one is single nucleotide variant. So this is just one change. And this is actually what the sickle cell anemia changes that we talked about. Just one tiny change out of 3.2 billion can cause that disease. Okay, another is insertion deletion. So you can just get a chunk of DNA added or a chunk subtracted. Another is a block substitution, where you just get a bunch of the same base in a row, and that's not what was supposed to be there. There's an inversion variant, where you flip something over, and this one's kind of interesting to think about identifying. We program computers to do all of this processing of the DNA sequences, and this is an interesting one for the computer to be able to identify. And then the last one is a copy number variant, where you can get an extra piece of a whole section. And that can affect, the dosage of an amount of protein that you have, or things like that. So then once you identify the variants, you say, okay, this area is not normal. You want to understand, what does it do for the person? For a lot of the variants, either we don't understand them, or we believe that they don't really necessarily matter. So again, we think about how the DNA gets copied into RNA and copied into protein. So we have a few different ways of classifying variants. Even with those single nucleotide variants, even if they're inside of a gene, they can have very different effects. So a synonymous mutation is when actually it doesn't have any effect on the downstream protein. It's one change, but it doesn't matter. A missense means that you just sub in one new amino acid, so it's one of the pearls on this chain. And so that could have varying effects. It could change the whole shape of the protein, or it could be very minor, depending on exactly the part of the protein that it's part of. And then the last one is a nonsense mutation, which actually means the protein stops early. So you could end up with half your protein chopped off. Again, I just think this is amazing. From one single nucleotide change, you could end up with half your protein gone. And then outside of the coding region, there are a lot of different methods in development. I hope see if this works. Okay, great. So this is actually an area of expertise for the group that I'm in, and this is a little complex, but I wanted you to think it's complex. We said only 1% of the genome is part of the genes, and then there's 99% of the genome that's outside. And there are many different things that happen in that part that it's an active area of research to learn what happens. So there are regulatory areas that might change, regulate up or down whether a gene is expressed. They might also affect how a protein is degraded, whether it stays in your blood for a long time or is eliminated. And there are just a lot of different things that can happen in the non-coding region that we really still need to learn about to be able to fully interpret. And then the last is copy number change. So I mentioned that on the last slide, where you can have a whole chunk where you have either an extra piece of that chunk, or you just completely lost the chunk. And this is a plot of this from a cancer patient. So on the x-axis here is all the chromosomes. Green is where all the copy numbers are normal, and red is where the copy numbers are abnormal. So in a lot of cancer patients, you actually end up with extra copies or too few copies in a lot of different areas, just because of different mechanisms that cause cancer to arise that we can talk about if anyone's interested. But these actually can be really important, because you can think, if you have, I don't know if you can see this, but there's some little spots on 10 in the middle, if you can see it like here, where you have the red dots that are way below. So that means you actually could be completely missing that protein. You could have 0 of it. And so you can think about, if that's something that's actually causing your cancer, you could just give that protein and it might be able to treat the cancer. Alternatively, sometimes you have 40 extra copies of a specific gene. And if you can stop that protein from being formed, and that's what's driving the cancer, it's also a way that you can treat the cancer. So these can be really great drug targets. Okay, that's the end of that section. Any questions on that one? Yes?
Your reference that you mentioned, does it come from the Human Genome Project? Or?
Yes. So basically they made the first reference genome as part of the Human Genome Project, and now they're on, I think it's version 38. So they keep updating it based on new information that they have.
How did Lumina end up with the monopoly?
They just have the best technology.
But it's not like the process in any way is proprietary. So
it is proprietary, yes. So they have their own mix of different chemicals that they put together, their own machines. When you buy the machine, they say, if you use any different mix of chemicals than what we give you, it invalidates your warranty. So then if anything breaks, you're out of luck. And everything's patented, and you're not allowed to reverse engineer it. But it costs a lot of money to do the research and development that went into making the technology.
It's totally contradictory to the purpose of science.
It's one of the challenges, definitely. You want everything to be open so that more advances can be made, and certainly a lot of government money goes directly to Illumina because that's what people need to do their research. And there are some competitors that are up and coming. There's one called Oxford Nanopore that makes a sequencing technology where the machine is about the size of your phone. So the Illumina sequencers are maybe a little bit larger than a mini fridge, but this new technology is this big. It's really amazing that it can do anything, but the error rate is about 30%. So they've got to bring that down. But they've been getting better and better every year, and those machines are extremely cheap. So a lot of people think that they might surpass Illumina in the next couple of years. And that would certainly, that could bring the cost down by an order of magnitude, which would really help open up the field. Okay? Yes.
So 1% of the genome you mentioned, did it have certain types of characteristics in it, or?
Yeah. So a lot of it is repetitive regions where you'll get the same sequence over and over. Sometimes it's like 3 nucleotides in a row, many, many, many times. And so what happens is the length of the repeats is longer than the length of the piece of DNA that you sequence. And so you're just not able to resolve where to map that puzzle piece because of these extra repeats. So there's long read technologies that are up and coming that are orders of magnitude longer than the 150 base pairs. Oxford Nanopore is one of them. There's another one called PacBio, where the cost of sequencing a genome is about $40,000, but the quality is excellent. So if you really need to map a small region, you can do it with the PacBio technology.
Clinically relevant? Did you have
Sure.
an example of which?
Sure. There's, if you've heard of ALS, I don't know if people remember the Ice Bucket Challenge, was raising money for ALS. So the number, I was actually thinking of that with the triplet repeats. So actually people that have extra expansions of a certain triplet repeat are much more likely to get ALS. And so it can be, first of all, they want to understand how many repeats people have with ALS versus not in the same family members. So it's kind of a research question, but in the future it could be, used as a diagnostic if we ever have a drug that we can use to help treat the disease. But that's one example. So the last part is about getting to what the team that I'm part of currently does to try to expand the use of genomics in healthcare. And then I also wanted to talk about open questions in the field, such as making sure that patients understand what's happening to their data. So a study that we have recently taken on is a cancer-focused study. And so it was actually announced as part of an Obama Precision Medicine Initiative Summit. It was a partnership between IBM and the New York Genome Center. And what we did was sequence 200 advanced cancer patients. So we basically wanted individuals who were candidates for experimental treatment options. So they were about to fail or had failed their first round therapy, but they were still healthy enough to be seeking treatment options. So we did across all different cancer types. And we had 8 different institutions collaborating with us. So this was a big effort run through the New York Genome Center with many different hospitals around New York. And the reason that we collaborated with all these institutions is because we didn't see patients ourselves. And so we had to collaborate with physicians who actually see the patients to get them to talk to the patients about whether they would want to enroll. And then we also wanted the physicians to be able to incorporate any information that we returned that they found useful into the individual's clinical care. So we have now returned results to 193 of our 200. We're almost done. And we found a lot of actionable variants. I would say in over 50% of the patients, we found things that we thought could be used to be incorporated into their treatment. And so what we did was we had a phone call with every single physician on every patient to discuss with them exactly what we found. And so educating the physicians was a big part of this for a lot of the hospitals. There are some hospitals that are research hospitals that are very advanced, and the individuals we were talking to knew a lot about it. And then there were some more outlying hospitals, like in the Bronx, where they don't have access to this kind of information. And we were really teaching the physicians for the first time why we were associating an individual's mutations with a specific drug for that patient, and they were considering incorporating it into their care. So the next step that we're doing is figuring out, did any of the patients actually take the drugs that we recommended? And if they did it affect their prognosis? Was it any better or any worse than anybody else's? And that's actually a multi-year process because, thankfully, these people aren't projected to die next week. So that's basically the outcome that you get is what's the survival time. And so in 5 years or so, I can come back and let you know how it went. But it was really thank you so much. Part of it was just a proof in principle to say, can we get the tissues from the patients? Can we sequence them in a time frame that's clinically relevant? Are the physicians actually going to get on the phone with us to get the results back? And all of that part, was worked out and went really well after we worked out some kinks. So this slide is about the kinks. Okay. So the first thing that we found was that the institutions often had restrictions around what data they could share with us. So we wanted the individual sample, and we also wanted clinical data that would help us interpret the genomic data. And a lot of times the institution has policies either based on their wanting to own the data or, I don't know if that's my hair, sorry, or based on just legislation around what information they're allowed to share or not. And many times we found that this was counterintuitive to what the patients would want. In some cases, we had direct patient contact. They wanted us to have all of their data, but because the hospital's policies counterindicated it, we weren't always able to get it. Another thing we found was the importance of pertinent negatives. So this is basically, we started by returning everything that we said, these are all the drugs that we think are suited for your patient, and this is why. And the patient would say, well, what about this drug that I was thinking about? Why didn't you recommend this? Did you see anything that would make you think that this isn't a good fit for my patient? And so we started to include information like that that said, these drugs are not as good of a fit because of some specific mutation. There are also different genetic markers that are basically markers of how good the person's outlook is that don't actually affect which drug they would take, but it would affect the way that the physician would manage the disease in other ways. And it might be something that they would want to communicate to the individual, based on your specific case, it looks like you have X or Y outcome. And then the biggest challenge is how the individuals actually get the medications. So there were many cases where the physician was on board with treating the individual, they talked it over with the individual, they wanted the medicine, and the individual is not able to get it. So one issue is even if the drug is FDA approved, sometimes the insurance companies are not willing to pay for it. And this is in part because genomics is such a fast-changing field that the insurance companies can't keep up with how many different genetic associations there are with particular drugs. And there's new logic coming out all the time that maybe it's not directly the protein that attaches to the drug that we are seeing the mutation in, but maybe it's one that attaches to that protein. And so there's a little bit of an extra step of logic that the insurance company might not be familiar with. And this is really going to be an ongoing challenge for us. There's actually currently legislation along these lines also being proposed in Congress to have individuals have what they call the right to try. So this is basically when a drug is in development, going through the FDA approval process, they try it on a small number of individuals. And if the clinical trial that they're trying it on these individuals on is not located near you or is just small enough that you couldn't get in, you're not able to get access to the drug. If you are able to get access to the clinical trial, by the way, it's free, which is awesome if you can get it. But if you're not able to travel to that site, it seems, in some ways it feels unfair that individuals that happen to live in the city where the trial is get the drug and the others don't. And so there's legislation out there right now about individuals having the right to try a drug that suits their condition. One of the problems with this is first of all, we don't know all the risks. That's the point of having a clinical trial and trying it on a small set of people. And also the companies that are making the drugs haven't set up to manufacture it for everybody. They've only set up to manufacture it for a small amount. And so they're concerned about the financial considerations and who might pay for the drug. Again, it goes back to the insurance company issue. If the insurance companies aren't paying for it, the pharma company can't give it away for free to everybody. So I think this will be an ongoing issue where individuals are well suited for a drug, but it's not really clear how they might get it. Okay, so next I want to talk to you about a new effort that we are just launching to use genomics in healthcare. So this isn't in cancer, but we took a lot of our lessons from the cancer study and incorporated into this, this new study that we are just starting at Rockefeller University. So what we want to do is crowdsource individuals to participate in our study. And so the idea is that it's anyone that is just a healthy individual, or not healthy, that's fine too. But basically anyone can enroll. And we look at everyone's genome to see what things we can find in it that are clinically actionable, similar to the examples that we talked about in the beginning. So the idea is that we return anything to the individual and their healthcare provider that's clinically actionable today, and then we study the impact of the results. So I'm going to go into a few more details about this study and our thoughts. So the first part is to crowdsource recruitment. So basically this came from trying to get around the issue where the patients want us to have the data, but the institutions are not able to give it to us. We want to directly engage the individuals and have them supply the clinical information. And we're hoping that this allows us to get more clinical information and have ongoing connection with individuals. And we've actually found in some cases that the individual is more motivated to get you correct data on something like family history than a physician might be, because they only have 15 minutes to see the patient and they're writing down everything they can. But if an individual is really motivated about figuring out a medical issue that they have, they might be able to more carefully go through the clinical data and supply it to you. We're also aiming to recruit participants that match the diversity of the US population. So this is an ongoing issue in scientific research. I just want to show a pie chart of the US population by ethnicity from the 2015 census. And this is the ethnicity of individuals that participated in genetic studies in 2009. So people saw this and said it was horrible. We have to really, try harder to have diverse populations participating. And it looks like maybe they did better in 2016, but actually almost all of the growth was in Asian populations. So now the Asian populations are overrepresented, and we even have less African populations than we had before. And so this is an ongoing issue that we still haven't really figured out how to solve, but we, at least in New York City, hopefully we can get a diverse enough crowd that can match the diversity of the US population. And this is important for a lot of different reasons. So first of all, it's fair because as we talked about before, what's the average genetic makeup in a person of one ethnicity is not the same as in another ethnicity. So it's important to know what the diversity is for each individual population so that you can interpret things correctly. Also, we found that since some mutations only occur in some populations, it's better even for scientific discovery because you would never make the correlation between the connection between a certain mutation and a clinical outcome if you never see it. And you have the chance of seeing more of those differences if you have a more diverse population. So the next part about enrolling individuals is about informed consent. So this is what I was talking about before, where you sit down with each individual who's interested in joining the study and you go over with them all of the risks and all of the benefits to their potential participation. And you actually have to make sure that they understand. You do something like a teach-back method where they explain to you what you explain to them, or you have an online quiz or something like that to make sure that the person really understands it. And often we have an informed consent form that often we translate into different languages to make sure that everyone can participate. And the individuals that actually do this process are usually genetic counselors. This is a really interesting role where you get to be an expert on genetics and explain it to the individual that's going to be participating and make sure that they understand before they agree to participate. And often we have different checkboxes where they say something like, can my data be used in future research? Can my sample be used for future things? And you can say yes or no to that based on what you're comfortable with. So then once the individual consents, they agree to participate. We obtain a blood sample from the individual that we can use for DNA sequencing. We ask them to fill out a clinical data questionnaire, and we ask them to give the information for their healthcare provider. And as I mentioned, the clinical data is critical for identifying the links between the differences in DNA and the medical outcomes. And healthcare provider information we like to include so that there's an individual that can follow up with them if there's something that's relevant to their care, because if we scale up this study, we're not going to be able to do that for everyone. So we want to be able to have a way to work it into the system that already exists. So then the next thing we do, we sequence everyone's DNA, and then we return actionable results to them. So we inform both the individual, if they want the data, they get to decide, the individual and their physician about anything that's a clear clinical action, like the examples that I mentioned in the first part of the talk. So right now we have a set of about 20 actionable targets that are incorporated into our current analysis framework, and we know that there are hundreds out there, and we're just working as fast as we can to incorporate all of them in. And this is another case, like I talked about in the cancer study, where educating the physicians to make sure they understand this information is really critical, and I think it's going to be a societal challenge coming up as genomic information gets incorporated into healthcare more. And this is another place where genetic counselors can help fill the gap. So actually, if a physician doesn't have time to explain it to the individual or doesn't understand, genetic counselors are well trained in this and can meet with the patients and explain to them their results. So then the next thing we want to do is expand our list of actionable results. I mentioned we have 20, and then we're going to have 200. And so what we want to do is, once we have someone's sequence, we can just reanalyze it every time that, one, we add new information, and two, a new discovery is made. So hopefully there will be many discoveries throughout the scientific community that we can incorporate, and then also we want to use our database to make new discoveries that we can add as well. And then the final step is to understand what actually happens to this information. So we want to maintain a connection to the individuals that enroll so that we can get more clinical data from them over time to understand, did they actually use this information and did it do anything for them? So the types of questions that we would like to ask are along the lines of understanding what are the costs and benefits both for the individual that's part of the study and for we call it the payer, the insurance company that's paying for it. And we hope in the long run to be able to convince insurance companies to pay for it so that the individuals don't have to pay, because again, then it would favor affluent populations and only they would get access to it. So these are the types of questions that we would like to ask. How frequently are negative events avoided, like the side effects with drugs that we talked about? Are follow-up procedures costly? A mastectomy is not a free surgery. Do the information cause and or relieve stress? So some people would be really stressed to find out that they have an increased cancer risk, and maybe they would rather that they didn't know. That's an important thing to be able to track in the population and see how people feel about getting this information back. And then finally, were family members tested as a result? We talked about the cascade screening with FH before, where you're looking at those second and third degree relatives, and this is, would increase the value of testing each individual. Okay, so I mentioned we're still figuring out how to enroll diverse participants. It's a challenge that is throughout scientific research. Challenges of obtaining informed consent, making sure that people understand what they're agreeing to, even though the field is changing and it's hard to know exactly what would happen to the data in the future. Returning data within or outside the healthcare system. There are a lot of companies, 23andMe is one where it's just the individual, they don't involve their healthcare provider at all, and some people favor that option over incorporating it into the healthcare system. Whether we return only actionable information or clinically relevant information, there are risk factors we know for things like Alzheimer's, where we know that it increases your risk to about 1 in 3, but there's not much you can do about it. So we've chosen not to return that information, but some people might want to know that because they're very interested. And then who should be able to access the data? There's also legislation being proposed right now about employers being able to access genomic data. What about law enforcement? Something that we can protect against in clinical research, but it's very challenging for for-profit companies to prevent. And then some more regulatory challenges. We can skip through this. And data security is another thing to think about. Okay, I can take questions. I just want to thank my boss, Bob Darnell, and Mayu Frank, who runs our clinical studies. If anyone happens to be interested in participating, you're welcome to email Mayu, and she'll give you some more information. Okay, now we can take questions. Thanks. Yes. Ooh, sorry.
So crowdsourcing your data
Yeah.
is one thing, but is there any kind of initiative within your group to kind of provide aid and resources to people who want to adopt the methodology of your study, like as a whole?
Oh, absolutely. So we'll definitely publish on it when the time comes, and there's a lot of scientific literature out there. For the most part right now, we're using other scientific tools that other people have published and just kind of piecing it together in a new way. But yeah, absolutely, there will be an effort to publish it. Yeah. A question in the front?
What is one weakness currently for reference genomes?
Oh, the reference genome. So one weakness is that there's only one version for everybody. It's not different for different ethnicities, which they're working on developing.
So there's only one kind for each person?
Yeah, one kind for everybody. And then the other weakness is 1% of the genome is missing.
Okay. Thank you.
Yes.
What qualifications do you have to be a genetic counselor? Like what kind of degree?
Yeah, it's a training program. I think it's after a bachelor's. It's a 1 or 2 year training program. And there are certain certifications that you get, and actually it's one of the most rapidly growing fields, and they cannot train people fast enough. Everybody wants genetic counselors. I'm guessing it's going to be in high demand for a long time. Yes.
How did you get into genetic counseling? You got a bachelor's degree in that. How did you go into that?
Oh, for me, I came to it from the computational side, actually. So I started analyzing large-scale data sets, and then I just happened to want to use my analysis skills and my MBA at the Genome Center, and then I picked up all the genomics stuff along the way. Basically, I wanted to do something that would make a difference in society that I felt was, helping and improving people's lives, and so I picked up all the genetic stuff after. Other questions?